and it has done that now. So here we are recording. I'm going to go ahead and fill the monitor with the slide and introduce the cell theory. All living things are made of cells. All cells come from pre-existing cells. This is true for everything we observe today and all of the evidence that's left by what I'd call a remnant. So when we study coal and the places where it preserves the imprints of living things, when we study fossils, when we study the formation of sediments in marine environments today, in the presence of life or in the absence of life, in the chemical traces it leaves, we find similarities between the cellular structures that are alive today and the chemical residues that they leave, and the chemical signatures in rocks in those environments. Life is going to be completely about what cells are and what they do. Remember, it is the cell in our body that is the living unit. It accounts for our growth. We start out as a single cell, as a single fertilized egg. What a remarkable thing. There's many multicellular organisms that arise from the fragmentation of their bodies. So cut off a piece and you can get a whole new organism. Starfish are that way. Tear a starfish apart. If you've got part of the central canal, it can grow four more arms or 3 more arms and live as an independent starfish. Plants do this, vegetative reproduction. Animals, we don't do this in our body. Growth, reproduction, tissues, organs, and organ systems all form from cells. In this chapter, we're going to explain a major disease based on cells. Cancer is a problem of the cell. It's a cell gone rogue. And so it's useful to understand cells, what they normally behave like, and what is the change that produces this tumor, this condition we call cancer. First of all, in our body, there's a small collection of cells called germ cells or sex cells, and these are our reproductive cells. In males, they will form the sperm. In females, they will form the egg. Those cells are different from other cells in the body. The rest of the body cells, and by the way, including the organs which are the reproductive organs, form the soma or the somatic cells. Except for the sperm and the eggs themselves, all of those cells are exact genetic copies of the fertilized egg. They are uniform from the time of fertilization, of conception, and they are multiplied in huge numbers to form our body and then differentiate into our tissues. Here's a cell diagram. We tend to look at the nucleus first because it's inside the cell. And that nucleus is bounded by a membrane, but why start there? It's the outer boundary that's important. I'm going to get my pointer back up so I can show you this outer boundary, the cell membrane or plasma membrane, separating the outside from the inside, or separating non-living from living, separating the environment from the cell or organism. For most of Earth's history, life on Earth, an organism and a cell were the same thing. All life was single cell. Multicellular organisms like us are sort of come lately in the evolutionary history of the Earth. Within this cell membrane, you have a complex cytoplasm, and the cytoplasm is a cytosol. This background that's sort of tan colored is a solution, a chemical medium that provides both the physical structure and support in the chemical environment for the operation of all of these little structures: the nucleus, endoplasmic reticulum, mitochondria, and Golgi apparatus, the fibers that we call the spindle or the cytoskeleton. Down below, we're going to see an endoplasmic reticulum that's covered with ribosomes, and therefore is rough ER. These are vesicles of particular types, little compartments of specialized chemicals that contribute to our metabolism. This plasma membrane, where we started, is peeled back here to show you a couple of features. It is a sheet, a barrier between the outside and the inside. It is formed by a phospholipid bilayer, but that's just the beginning, the hydrophobic zone that separates it from the hydrophilic environment that surrounds it, the pond water, if you will. Embedded in it are proteins, are channels, are chemicals that allow this membrane to be selectively permeable. To maintain life, you have to have an intact membrane, and to maintain life on an ongoing basis, you must exclude the things that would interfere with your metabolism and obtain the things that are required. You must, from the inside, recognize the waste that would poison your metabolism from within and transport them out. The other thing this peel back shows is the fibers of the cytoskeleton. We used to think of this as a bag, as a kind of a plastic sack that is just held out by pressure from the liquid within, but it's not. These microfilaments and microtubules are fibers that are constructed and form a kind of an outer shredded wheat-like shape that the membrane drapes on, and that provides the ability to form the cell with space inside, to grow the cell, and then this cytoskeleton needs to be torn down when you're going to divide the cell and create two cells. Here's the nucleus, a double membrane organelle that is separated from the surrounding cytosol. These important nuclear contents are the chromosomes, the DNA that code our genes, and the mRNA that is carrying the message about our proteins out into the cytoplasm. Here is an electron micrograph. Now, a light microscope like you need in lab can only magnify 1,000 times. You get much greater resolution by using electron beams, and here you can see so many remarkable things. Here's that dark staining nucleolus. That is rRNA that is being produced in the nucleus to form ribosomes. This double membrane, you can see clearly that membrane's a double membrane, and at various places where you see the arrows, you can see the pores in this transmission electron micrograph. Here's another thing that shows you the pores. This is very interesting. What you do is drop the cell tissue into liquid nitrogen and freeze it hard like a rock, and then you hit it with a hammer. What that causes is the fracturing of those solid materials, and then you look for places that tell you what the structure is. This is the outer surface of the nucleus. Do you see all the imprints of the pores? Now, when you shattered it, because the nucleus is a layer right next to cytoplasm, you basically broke the cytoplasm away, revealing this outer membrane. Here's a place where the nuclear membrane broke, right along where the pointer's traveling, and what you're seeing on the left side, upper left-hand corner, is the inner frozen surface of the nucleoplasm, and on the surface you can see the imprint of the inner surface of those pores. This is called freeze fracture. And this image of the nucleus is magnified 9,000 times. Here's the real barrier, the real important starting membrane. You cannot have a living cell without it. This is the phospholipid bilayer. Those micelles that we saw later were spherical, but this is a double sheet. Do you see this sheet of hydrophilic heads and how the hydrophobic tails are all below them? And this reversed sheet with the cytoplasm facing the hydrophilic heads and the hydrophobic tails above them. Combine them with the cholesterol, these yellow squiggly lipids that are loaded into membranes, and what you have is a surface, a sheet, with a hydrophilic outer surface for the environment and the water environment out there, and a hydrophilic inner surface for the water-based cytoplasm that is holding together this hydrophobic zone. This hydrophobic zone is something that electrolytes and hydrophilic molecules cannot penetrate. So we have to have special adaptations to allow for the transport that is needed. Sticking out here are glycolipids, providing perhaps binding sites, particular places where things adhere. Some of our disease pathogens, like bacteria or even our viruses, might be harmful because they have a binding site, bind here, and they can be working their way through the membrane to infect the cell. We see other features here. Here are the microtubules and microfibrils of the cytoskeleton. Notice how we throw up these fibers like structures and then we glue them together. These provide anchoring points for these long protein tails that wrap around them, stabilizing these protein channels that we see embedded and accounting for some of the specific transport, the semi-permeability of this membrane. Everything that happens on this membrane is very specific. If there is a need to transport potassium in, because potassium is a +1 ion, there has to be a channel, and that channel is typically a potassium-only channel. Here is an electron micrograph at 30,000 magnifications, showing where the pointer is going right now, the outer plasma membrane surface, but on the upper surface, there are these protrusions like little fingers or hot dogs sticking up and producing this hair-like surface. If you look very carefully, you see that the surface of those fingers looks fuzzy. And if we blew this up more, we would see the individual membranes are not just folded up into this tube structure, but the individual membranes wind back and forth, producing individual smaller tube-like structures. What I want to tell you is this is not just ornamentation. This means something because it's costly to build. You can kind of see in this finger right here how several fibers aggregate and basically move up, forming a column that this membrane is draped over. In other words, they're forming these projections on purpose. Now, what this does, wherever you see an elaboration of the membrane surface, by finger-like projections, by stacks like pancakes or coins, by folds, a membrane that goes back and forth and back and forth and back and forth. The purpose of that is to increase the membrane surface area. So this might be this is an intestinal cell in the small intestine where absorption of nutrients will occur. These are permeable membranes, and nutrients must move across the membrane to get to the cells on the lining. That nutrient absorption is hastened by increasing the surface area. This is a very different finger-like projection. Instead of for exchange, either elimination or absorption, or it is a structure that looks the same. It's a tube-like projection, but if you watch it on a living cell, it's waving back and forth. It's burning energy, and it's flailing back and forth like moving an oar. This is a cilium. And the cilium in our body that moves materials along inner pathways is structured the same way as the cilia and flagella in the microorganisms you studied in biology. You can see if you cut across here, you have this 9+2 arrangement of microtubules. On the base plate, the basal body is triplets of microtubules. See the 3? And it's 9 triplets with a hollow center. It makes no difference if you're a paramecium or a human being. Your cilia are constructed in this way. Now, given how complicated this is, what is likely is that it arose once, early on, and it was so useful that it was present in all the organisms we today call ciliates and flagellates. And as those cells through evolution gave rise to other forms and eventually multicellular organism, this is what we call a conserved structure. Something that performs the same function in all organisms where it occurs. Now, outside the nucleus, you get folded membranes. Again, this is more than just folding your pillowcase into a nice rectangle. Look how convoluted this is. Do you see how it forms these inner passageways, these kind of hallways that go up and down and all around the nucleus? Notice how they're connected from here to here and here to here. This is a passageway that's continuous with the inside of the nucleus. So we can use this space of the endoplasmic reticulum for metabolic chemical reactions. And in this case, because they're covered with ribosomes, these little black spots, we know that the principal job of rough endoplasmic reticulum is to produce protein. The ribosome is the site of protein synthesis. A gene on the DNA makes an mRNA that carries the code to a ribosome and assembles with it. And it is the ribosome and mRNA that then synthesize the amino acids into the right protein. This ribosome occurs in all microorganisms and multicellular organism alike. Now, look here where the rough ER gives way to smooth ER. Here is just no ribosome. So the protein synthesis is over, but further metabolic reactions, packaging, sequestering of chemical products in not individual molecules free in solution, but in collections, in droplets, is occurring here. And this brings up a really, really interesting property of membranes. They are barriers, but they're not rigid walls. They're not even impermeable plastic. They're fluid. They flow according to the cytoskeleton support under them and can be used to form compartments by bubbling off. I'll show you that in a minute. Forming vesicles. Vesicles then can move through the cytoplasm, and where they reach another membrane, those membranes can actually merge. This is something you can kind of see. If you put oil and water together in a jar and shake it up, you'll notice it gets cloudy, but when you set it down instantly, those oil droplets start hitting one another and merging into bigger and bigger and bigger droplets. And if you look for a period of time, pretty soon you have a layer of oil and a layer of water. And they're separate because oil and water don't mix. This hydrophobic property, the hydrophobic zone of the membrane, allows them to separate and form a separate bubble and merge and form a larger compartment. And this is the way vesicles are active in transport. Now here's a function. Here are some of those secretory vesicles, maybe formed, see them forming around the endoplasmic reticulum, but moving to a folded membrane structure that's separate from the nucleus. This is the Golgi apparatus, and it is a further compartment for specialized chemical reactions. I mean, you think about this. A cell that comes from cell division is a generalized cell, an exact copy of that fertilized egg that it started from. But in the human body, it will differentiate into a specialized cell. Perhaps it's a mucus cell that's making mucus in the cytoplasm and packaging it for secretion at the surface, at the epithelial tissue. So the subunits from endoplasmic reticulum that are protein would move by vesicles over here and merge, see this merging going on here, and dump their products into this space where metabolic reactions that are arranged on these membrane surfaces. Why do we need more membrane surface? Because these metabolic reactions have many steps. So we line the membranes up, the enzymes up, in a basically an assembly line, like a factory. So that if you get the reactants to the first enzyme, it's passed down the chain through reaction 8, 9, and 10, and product is produced. Once those products are produced, you can pinch off those vesicles. Maybe this is a secretory product like mucus. These vesicles then would move to the outer membrane, look at that ample surface area, and upon command, release their mucus into the space above this cell. Here's a kind of a summary of the things we've covered so far to kind of put some of this together under the heading protein synthesis. The double-stranded DNA, the double helix, forming the interphase chromosome, is here being transcribed into mRNA inside the nucleus. Here it's moving through a pore. Now, mRNA can grab a ribosome and synthesize protein right in the cytoplasm, or it can associate with a ribosome on the endoplasmic reticulum and synthesize that protein and dump it across into the ER space. If that is done, then these multiple protein molecules can accumulate in these endpoints in the smooth ER and bud off as a transport vesicle. The transport vesicle is transported. By the way, these bubbles are not just floating around. They can actually identify the cytoskeleton and move along it straight to the Golgi apparatus where they deposit their proteins. Other vesicles from other places might be introducing different proteins. And so by the time they are packaged and combined, moving up and back through these continuous spaces that are folded together, you get the packaging of different products, like this vesicle that's going to remain inside. A lysosome contains digestive enzymes. Out here is a secretory vesicle that is packaged a couple of protein products for this vesicular transport by merging with the outer membrane and expelling it. Now, if you work on the electron microscope, the magnification is so great that it takes forever to find something, but this electron micrograph is showing you this process right here. Do you see how the vesicle, the fixation, the freezing of this material, the fixation of it, immobilization of it, occurred just as this vesicle contacted the outer membrane. And you can see the pore that forms and the explosion as that pore opens, how the contents of the vesicle are shoved out. In the same way you can watch oil droplets merge like this, bam, and it happens faster than your eye can follow. And sometimes if you watch, it will kind of vibrate back and forth. There's a tension to that circular vesicle. So when you produce a pore, that vesicle membrane now is continuous with the outer membrane, the plasma membrane. And it basically goes like this. And basically that circle of membrane now was previously the membrane of the vesicle. So that material is not wasted. It is now part of the outer plasma membrane, because a membrane is a membrane is a membrane. Here we have the function of lysosomes that I mentioned as containing digestive enzymes. The ability of vesicles to enfold and capture material from outside, or to merge with the outer membrane and expel, are shown in this figure. Number 1 shows a process where a lysosome was released, and the lysosome has merged with a damaged organelle. It contains enzymes that degrade that organelle. There is some reabsorption. Imagine this. You're breaking down proteins. Any amino acids that are released can be reused. Any nucleic acids, any monosaccharides that are released. But in the end, you don't reclaim everything. There's still some waste. And so this mature, digested waste product merges with the outer membrane and expels. This rejection is called exocytosis. Number 2 shows you another thing where endocytosis has incorporated something. Now, this might be a nutrient to be digested. It might be a pathogen, a solid. It might be a poison. But once incorporated by endocytosis, see it being drawn in? A lysosome merges with it and breaks it down. So if it's a pathogen, it kills it. This is a great place to kill a bacteria. Once the bacteria is dead, it can't provide a noxious infection. There's some reabsorption as before, but always waste, and exocytosis will expel that residue for waste collection. We do see a third thing happening to primary ribosomes. Sometimes they simply burst and liberate the digestive enzymes into the cytoplasm themselves, where they act upon components of the metabolism that are free in the cytosol. Here's a fascinating organelle, membrane bound again, and the only thing I want you to notice first is the double membrane structure. This outer covering, like an outer balloon, enclosing this densely folded membrane that forms the inner membrane. I'll talk more about this later, but I first want to say to you, this means increased surface area. So I notice we're 10 minutes from the end, and I want to leave some time for questions. So I'm going to stop this recording at about 7.